Long before laboratories, Earth was already making mountains of sweetness in green tissue. In sunlit cane fields, sucrose rose quietly through tall, jointed stems. Beneath cooler soils, sugar beets stored the same bright treasure in pale roots. In orchards and forests, flowers poured sucrose into tiny drops of nectar. For plants, sucrose was portable sunlight, packed for travel between growing places. For people, it became crystals, trade routes, comfort, celebration, and temptation. Yet each sugar crystal held a small locked problem at molecular scale. Sucrose is two simpler sugars clasped together inside one neat molecule. Glucose and fructose are present, but joined beyond ordinary touch and taste. Water alone meets that bond, pauses briefly, and mostly passes by. Early cooks learned heat and sourness could slowly change a stubborn syrup. But nature had already found a quieter, more exact key for sucrose. In leaves, fruits, roots, and seeds, invisible helpers handled this abundant sugar. Yeast cells living on ripe fruit skins met sucrose as food. They could unlock it before drawing the smaller sugars inward for growth. Bees solved the same problem during a golden journey from flower to hive. At the flower, nectar was gathered as a fragile, watery sweetness. Back at the hive, bee enzymes reshaped it into dense honey. The hidden worker in that transformation was invertase, patient and small. Its name would come from a strange behavior of light in sugar solutions. Dissolved sucrose twists polarized light in one measurable direction inside a polarimeter. After splitting, the new mixture bends that light the other way. Chemists called the reversal inversion, and the enzyme became known as invertase. The first clues appeared in 19th-century rooms filled with careful glassware. Sugar solutions stood beside balances, lamps, notebooks, and narrow polarimeters on wooden benches. Scientists saw that acids could invert cane sugar without any living organism. Then yeast brought a more mysterious, more delicate change to the bench. In 1860, Marcellin Berthelot studied yeast extracts and cane sugar. He found a soluble ferment that inverted sucrose outside intact living cells. Berthelot called it a ferment of inversion, later remembered as invertin. The discovery was quiet, but it cracked a large 19th-century idea. A power associated with life could be separated from the living cell. The old boundary between chemistry and living matter grew thinner and more porous. Soon the word enzyme would describe a catalyst born from life. Invertase became one of the early witnesses for that new word. It was practical, dependable, and visible through the turning of light. By the late 19th century, Emil Fischer studied sugars with unusual care. Invertase helped reveal that enzymes were selective, not general dissolving forces. From such work came the enduring image of a lock and key. A molecule did not merely meet an enzyme, it had to fit. In 1913, Leonor Michaelis and Maud Menton chose invertase with purpose. They watched its behavior as sucrose changed, step by measured step. Their work turned enzyme action into a mathematical landscape for biology. With sucrose and invertase, researchers gained curves, constants, and prediction that still guide laboratories. From a sweet reaction came part of the grammar of modern biochemistry. At the microscopic scale, invertase is a folded protein landscape in water. Its surface holds pockets, ridges, and tiny charged resting places. A sucrose molecule drifts close, tumbling among countless moving water molecules. For a moment, the double sugar settles into the waiting pocket. The enzyme gently strains the bond between glucose and fructose. A water molecule is placed where it can finish the opening. The clasp breaks, and two smaller sugars slip away separately. Invertase remains behind, unchanged, ready to welcome another sucrose molecule. No flame is needed, and no harsh mineral bath is required. The work is small, repeated, and almost impossibly precise beyond sight. In the hive, that precision helps nectar become stable, fragrant honey. In fruit, it helps shape sweetness as ripening changes texture and aroma. In yeast, it opens sucrose before fermentation can move forward inside vats and doughs. In confectionery, invertase can soften fondant centers slowly over time. A firm sugar filling may become smooth, glossy, and gently flowing. In syrups and preserves, it helps discourage gritty crystals from forming. In baking, it can support browning, softness, and steady fermentation. In beverages, it helps create sweet blends that remain even and clear. In some fermentation industries, it prepares sucrose for microbes already waiting. Across green industry, the same reaction offers a milder route. Instead of forcing sugar apart, we can invite biology to help. Less severity can mean fewer unwanted byproducts and a lighter footprint. It teaches that enzyme history is not only speed, but elegance. It shows how small biological tools can guide large human systems. A future of cleaner processes may begin with such patient, quiet keys. Enzymes Bio exists because good science should be simple to source. We're enthusiasts who put the world's enzymes in one place for startups, labs, and B2B buyers. Kinder chemistry, here for generations to come. Find us at enzymes.bio.